There we go.
Actually, today I'm gonna need your help because I
Yep.
need to make sure that anyone who's online has been
All
that they
right.
can go into
They're asking questions. Okay.
Okie dokie. Okay, if you don't mind, I'm going to get going. We have a lot to cover today, and again, thank you for coming to the last class. I know it's been a really quick 6 weeks, but it's also been a long 6 weeks. So we've been looking at how synapses modify themselves with learning, and hopefully you've had the opportunity to look at both LTP as well as LTD, and I'm going to come back and revisit that in sort of the review session that I'll have at the end. So today we're going to be talking about the total destruction, not just of synapses, but also of neurons, sort of in the context of Alzheimer's disease, which I know many of you have heard about Alzheimer's disease, many of you have heard about Alzheimer's and dementia, but I also wanted to give you a slightly different perspective that when you hear things about Alzheimer's, or when you hear things about beta amyloid, or if you hear things about ApoE, which many of you might have, I want you to have a better understanding of what people are actually talking about. So I'm going to actually begin where many of you are kind of interested in going. So I didn't have a stellar undergraduate career. So I got into graduate school, and in graduate school I tried to compensate for all the things that I wasn't good at as an undergrad. So I worked pretty hard as a graduate student, to be honest. I always felt really guilty not doing well as an undergraduate student. So when I was in graduate school, I was often working like 20 hours a day in the lab. So many of you will pass by this building on your way to MSB. It's on your left, and now it's named the David Naylor Building. But when I started my graduate studies, this is where I would be spending like 20 hours a day, 7 days a week, basically sleeping and eating here. And this was where my office was, right above the doorway on the second floor. And I was involved in neurodegenerative research, and I was working on different forms of cellular apoptosis. That was a brand new idea. No one had thought of apoptosis being all that important. And I was interested in Parkinson's disease, Alzheimer's disease, and just generally understanding about this. So I was fully invested in doing my graduate research. I spent all my time in the lab, and I spent a lot of time there. And it was actually to my detriment. I actually didn't enjoy my research very much. I enjoyed being in the lab. I enjoyed figuring things out in the lab, but I actually didn't enjoy the overall experience. And that's something that maybe I'll talk about at the very end. But it was also bad in another way. If you're working 20 hours a day in the lab, 7 days a week, which I was doing, and it's not unheard of still to do that in these days, a lot of my interactions with my family actually started to erode. So I moved downtown. I was living very close to my research lab, etc. So I don't know if any of you know this individual here. Any ideas? Okay, that's actually very relieving because it's a family member of mine. So if you knew who that was, I'd be like, wow, that is so incredibly creepy that you might know my family members. So this is actually my grandfather who passed away. And my grandfather who passed away a number of years ago, he passed away while I was in graduate school. One of the things that my dad told me is that when my grandfather passed away, that he actually had a form of dementia. I was working on Alzheimer's dementia and different forms of cellular apoptosis, but to be honest, I really didn't care. And I know it sounds terrible, but I really didn't care that my grandfather, who I'd never met before, had passed away from Alzheimer's disease until I got into my PhD. And there, my dad also called me up one day while I was working in the lab, and he said, oh, by the way, your uncle who lives in Brazil, who I did know, who I'd met several times, your uncle also has Alzheimer's disease. This was many years later. He also passed away from Alzheimer's disease. So up until then, Alzheimer's disease was just sort of an intellectual exercise. I'm going to work on this disease because it might help me to get my master's degree. It might help me understand how to publish papers better, and it was something that I was doing as part of being a research student. It had no impact on me on a personal level whatsoever until I realized that many, many, many members of my family actually have Alzheimer's disease. So my grandfather passed away at the age of 65, my uncle passed away at the age of 61, and many other members of my family also passed away from Alzheimer's disease, and really bad forms of Alzheimer's. Many of you will have heard that Alzheimer's not just causes you to lose memory, but also to change your personality. And when they passed away, they had like the worst forms of Parkinson's. They'd be yelling at their family members. They had like full-blown loss of bowels. Motor control was disappearing. And it's a really scary disorder in that way. I also mention this because many of you have heard or will hear about my own struggles with depression. Now, I've had depression in the past, and I really hadn't realized that depression is actually a risk factor for developing Alzheimer's. And it's one of those other reasons why we're so interested in trying to prevent depression and mental health disorders in young people, because we don't know all of the mechanisms that are involved, but it does seem to actually impact things like Alzheimer's. So I'm going to tell you about some of the things that are going on here at the University of Toronto, but I don't actually want you to end up like me, where you're just doing things because it's a natural part of being a research student, or I have nothing better to do, or I'm not really sure what I want to do. I also don't want you to get too discouraged because, again, if I was able to get into graduate school and I was able to get a job in a position here at U of T, that ultimately all of you who are much brighter than I am have a lot of hope to do just as well or even better. And I also want you to understand that some of the things that we talk about in this course isn't just about this course. Sure, you need it to get a good mark on your final exam, but many of you, in fact, I can guarantee you that 9 out of 10 of you will be impacted, just as I was, by Alzheimer's. When I was a student in undergrad, it never crossed my mind that Alzheimer's would be a disease that may impact my family or my own personal life. So I also want you to think a little bit about this, those questions that I'm going to be asking you on the final exam, that I'm going to be preparing you for, that you also have this opportunity to make an impact on this disease yourselves. There is a lot of research going on here at the University of Toronto and across the UHN that actually deal with this, and I'd be very happy to help you with this. So I want you to also realize that you have to get a clinical diagnosis. I don't care if you're talking about a mental health disorder. I don't care if you're talking about Alzheimer's disease or Parkinson's. If you think you have multiple sclerosis or you think you're having a stroke, always get a clinical diagnosis, because sometimes it gets really, really confusing. Now, we're talking about Alzheimer's, and you might be thinking, wow, this case study, 55-year-old male, someone who's into the late part of their middle life, is going to be coming into the emergency room, and they look like they're really confused. They're not able to draw a clock normally. They can't tell you what time it is, and it gets progressively worse. But the reality is there are many forms of memory impairment. And again, we've talked about this a little bit and think a little bit about this cognitive impairment that might lead you to suspect that this is Alzheimer's disease. The inability to draw a clock normally might make you suspect, yeah, they probably have Alzheimer's disease because we're talking about that. But you also, again, need to have a proper clinical diagnosis. So I want you to think a little bit about this. This person also has swollen ankles, also has abdominal distension, and those of you who are in physiology, you might have a better diagnosis than having Alzheimer's. What do you think? Any ideas? So this person is middle-aged, late middle-age, also a prime candidate for Alzheimer's disease and or dementia. What else could they be suffering from? Yes. Could be renal failure causing toxic metabolites, but renal failure may not be causing abdominal distension or swollen ankles. Anything else? Yep, you could. But there's something else that's equally important for having swollen ankles. Anything else? Could be a protein deficiency? Yes. Could be heart failure. There could be a number of different diagnoses. In fact, all of them sort of revolve around the same thing. You can be totally confused and you can have all of these issues that you're seeing here where you have cognitive impairment or you look like you have cognitive impairment simply because you have congestive heart failure, if you have something wrong with your protein levels, etc. So I want you to, again, think a little bit about the clinical side of things. Again, not always Alzheimer's when you have cognitive impairment. So I'm going to give you a really brief overview of Alzheimer's disease, which you don't need to memorize. You're not going to be asked any questions about that whatsoever. Today is really to focus you on thinking about Alzheimer's disease in the future, but also to make sure that you know exactly what's expected of you for the final exam. We don't have much time, in other words, to make sure that you are going to do well on this section. I'm going to tell you a little bit about the gross pathology. Many of you have heard this before. I'm not going to be testing you on this, but I will be testing you on the generation of what is known as beta amyloid and why beta amyloid is really important. So at that point, I want you to be paying some more attention. Also getting into treatment strategies and why that's important for Alzheimer's disease, why we don't have good drugs for Alzheimer's disease, all of these things. And then at the very end, I'm going to ask you to think a little bit about cholesterol and a particular marker for cholesterol known as ApoE, and why ApoE, microglial cells, and astrocytes are important for Alzheimer's disease, and particularly late onset Alzheimer's disease, which means that you might get it when you're in your 60s or 70s. That's that late onset, not the early onset where some individuals are in their early 50s or even in their late 40s when they develop it. So the good news is the early projections that we had at the beginning of 2010, when people started to really worry about this, the early projections way back here in 2010 saying that we were about to face this tidal wave of individuals that are going to have Alzheimer's disease as we have an aging population. Yes, the numbers are certainly going to be rising, and they could be on the upper end or they could be on the lower end. But right now, as of 2017, we actually feel a little bit better that we're going to be on the lower end of things, not on the upper end, because we could be all the way up here or we could be all the way down here. And right now, it looks like we're more likely to be on the lower end of Alzheimer's disease, at least according to the Journal of the American Medical Association. However, it's also becoming very clear that the incidence is going up, and Alzheimer's disease is becoming much, much more common. And the last full study that was done at the end of 2010, we have new data coming out that should be ready in about 2 years' time. We'll know a little bit more about this. We know that Alzheimer's disease actually has been increasing. Some of you might argue that it's actually likely that there's a better diagnosis for Alzheimer's disease, or we have better detection methods, whatever it is. The incidence of Alzheimer's disease has gone up dramatically, 66%, versus other things like stroke. The incidence, again, may actually be plateauing off. And we've talked about some of these issues before. Many of you will know some of these different symptoms that are associated with Alzheimer's disease. Many of you will associate Alzheimer's with memory loss. And to be honest, one of those things, and I'll share a little bit more about my family history in a few moments, one of those things that really, really bothers me and makes me even more anxious is when I have a really bad memory day, because all of our memories get affected, and you could have congestive heart failure, etc. But when I have a really bad memory day and I can't remember a person's name, someone who I've met with like every single day and I can't remember their name, not that I can't recognize their face, but I can't remember their name at all. It actually really bothers me. And this memory loss that we associate with Alzheimer's is important, and I'll tell you why it's important in a moment. But there are so many other things that are also affected in Alzheimer's disease. We know that attention and being able to pay attention, because the nucleus basalis of Maynard is also affected, the cholinergic projections at the base of your brain. We know that attention deficits also arise. We know that as a result of those connections being affected, that there's a lot of problems with planning as well as solving problems, which is why a neurologist will ask you to draw a clock and to see if you put the right numbers on the right positions of that clock. We know that there's also a lot of confusion, spatial disorientation. Many of you will have heard of individuals who go out who have Alzheimer's disease and then end up in the news because they can't find their way home, and there's this lost senior citizen report out there. Again, lots of changes in mood as well as in personality, which is a scarier thing if you think about that. Lots of problems with communication, difficulty finding words. So some of you will know that if you come to see me, oftentimes I'm kind of off trying to figure out what the next word that I want to be able to say is. And those are things that, again, might be some things that you may not have associated with Alzheimer's. It's thought to be one of those earlier signs and symptoms. Do any of you know what sundowning is? And again, you're not responsible for this slide, but sometimes I think it's just worthwhile learning about something for the sake of learning about something. Sundowning, have you heard of it? Yes, at the back. So your colleague is 100% correct. As the sun goes down, as it gets dark outside, some individuals, not all people with Alzheimer's disease, but some individuals will actually exhibit increased agitation. They get more nervous, or they get more disoriented, or they get more confused, especially as the sun goes down. So if you hear that phrase in whatever classes you're taking next, hopefully you'll be able to relate that back. So again, the gross pathology and the different areas in which this starts, we know that there is overall shrinkage of many different areas within the brain, but in particular within the medial temporal lobes where we find the hippocampus. This is one of the areas that is particularly vulnerable to Alzheimer's disease. We know that as the brain starts to shrink, the areas in which we normally find the cerebral spinal fluid, the ventricles, actually start to appear larger and larger. And again, that form of brain atrophy that's accompanied by the enlargement of the ventricles is something that is quite severe. Again, there is this pronounced shrinkage of the hippocampus, which again is thought to account for the memory loss, some of the problems with decision making, etc. And again, this brain atrophy is believed to, in many cases, but not all, but in many cases, to start off in the medial temporal lobe, specifically within the hippocampus, and it will then spread from there into other areas like the association cortex. It will also affect the sensory and motor areas of the cortex as well. It also starts to affect, as I mentioned to you earlier, the nucleus basalis of Maynard, which is a cholinergic projection that's found on the base or the bottom of your brain. And this projection is thought to be really important for controlling sleep, for attention and consciousness. And as you start to lose more and more of the synaptic connections first, and then these neurons start to die and become atrophic, it makes sense that you're going to lose concentration. The sleep cycles will be reorganized, as well as not having the appropriate sleep-wake cycles. And again, there are lots of pharmaceutical agents that have been brought into play that target the cholinergic neurons to increase the levels of acetylcholine within the brain, especially as we start to lose them. Because we're dealing with a course on synaptic physiology, I thought it was really important to emphasize to you that one of the first things that we think happens, we actually haven't been able to see this because it's really hard to visualize synapses, and it's perhaps one of those things that as you're going into other courses and you hear about structural imaging studies, and you might hear about diffusion tensor imaging or other newly developed forms of MRIs, that we actually can't see at the level of a synapse. That is far beyond our capabilities right now. It's a lot like saying we could go to Mars, but the reality between saying we could go to Mars and actually going to Mars is a lot like being able to visualize synapses at a structural level in an MRI. We are not at that technological stage quite yet, but it would be really important for us to know if synapses are being lost, especially within those cholinergic projections. We also can't tell, although we're getting better at this, and some of you will hear about PET, which is the positron emission tomography. We have drugs that we think now are able to pick up increased levels of extracellular amyloid beta deposits, and we think that we can start to detect this early. There are also other tests that might be in play to be able to detect neurofibrillary tangles as well as tau proteins within your cerebral spinal fluid in the early stages before you develop the symptoms of Alzheimer's disease. We know that there's also going to be the glial scarring that accompanies the loss of neurons, and more importantly, we also know that there is an immune response. I want you to keep this in mind, that when we look at the brain, we've been really concentrating on neurons, but of course glial cells are really important, and the immune component of the immune response in the brain always involves the microglia. So when you're asked, what are the glial contributions? We have contributions from astrocytes or glial cells that form these scars, as well as the microglial cells that might be involved in the immediate and early immune response. So I'm going to switch over now into what we think is the pathology. There's a lot of debate about this. Considering that we have known about Alzheimer's disease for well over a century since Alois Alzheimer first described this in his patients, and he was able to examine the brains of these patients for over a century, it's really surprising that we actually don't know which of these two things is the chicken and which is the egg. We don't know, and there's no consensus, because every year someone else comes up with a different hypothesis. Is it extracellular beta amyloid that causes Alzheimer's disease, or is it the accumulation of these neurofibrillary tangles and tau that causes Alzheimer's disease? Those are contrasting theories right now. There is no clear-cut winner. It's really split exactly down the middle as to which one starts the process of Alzheimer's disease. Is it beta amyloid, or is it neurofibrillary tangles and the tau protein? I think I just heard Siri or something turning on. So I'm going to take you through this. These next few slides, I want you to tell me if there's anything that you don't understand. I'm going to be really straightforward and honest with you. One multiple choice test question and one of your short answer questions involves these next few slides. So what we're looking at is, as the brain starts to atrophy, what causes this atrophy? Is it these amyloid plaques, or is it the inclusion of these neurofibrillary tangles? Which one causes it? We don't really know. And I'm going to take you through some of the evidence that we think might actually be important for one versus the other. In particular, for the beta amyloid theory, it's also known as the beta amyloid cascade theory, that this is what begins Alzheimer's disease. That somehow these neurons begin to accumulate these extracellular aggregates that are well described within the pathology of Alzheimer's disease. If you look at a brain of someone who has initially been diagnosed with Alzheimer's, the definitive diagnosis is the accumulation of these beta amyloid plaques. We believe that these plaques that have lots of this protein known as beta amyloid, or A-beta, that these plaques somehow induce cytotoxicity. They're the ones that are killing off these neurons. They're the ones that cause the disintegration of these synapses. They're the ones that disrupt normal neuronal function, and they affect ionic concentrations. They affect the ability for the neuron to produce action potentials. This is supposed to be the smoking gun. I'm going to tell you that this makes a lot of sense, because we have this accumulation on the outside of neurons. In many cases, we have some models that suggest that this happens, and it also makes sense that all of the cells that you have in your body, and in particular neurons, have what is known as amyloid precursor protein. It is a normal protein that is found on your neurons. It's found on the neuronal membrane, just like an ion channel is, and I'm going to show you a diagram of this in the next slide. And this amyloid precursor protein is something that we have in abundance within our brain. We have it. It's normal. We need it, actually, as part of the normal brain. However, we know that as you get older, or after an insult, or after inflammation, that we can actually process amyloid precursor protein in two different pathways. One, we have what is known as a non-amyloid producing pathway. And again, if amyloid causes Alzheimer's disease, this is the pathway that would actually be better for your brain to take this precursor protein and make these non-amyloid fragments from this. So this involves alpha secretase as well as gamma secretase. The amyloid pathway, and again, the name comes from beta amyloid, comes from the fact that it is cleaved first by a beta secretase. That's why we get beta amyloid. The beta secretase is involved, as well as a gamma secretase, the same gamma secretase that's involved up here. So whether or not we have the alpha pathway or the beta pathway becomes really, really important. You need to drive things from the beta into the alpha. If you want a nice healthy brain and you don't want a lot of beta amyloid deposition, you want to make sure that the alpha non-amyloid pathway is the one that dominates within your brain. Do you have any questions on this before I get going into the next series of slides? Katja, you had a question earlier on and I sort of interrupted your thought process. Do you have a question? Okay. So this is a normal membrane protein. And this is what amyloid precursor protein, and on your exam you may also use this abbreviation of APP. This is the normal topology, and again we've talked about topology before. There's this large N-terminal fragment, it crosses and has a transmembrane sequence, and then there is this short C-terminal domain. And this sticks outside of the normal neuronal membrane as you see here. So I'm going to take you through the non-amyloid pathway first, and then I'll take you through the amyloid-genic pathway in a few moments. So this amyloid precursor protein is normally targeted. It's part of its normal life cycle, because remember, membrane proteins like this normally undergo degradation. This is normally cleaved by alpha secretase first, and you can see alpha secretase will make a cut here, and as a result it forms two different fragments. This fragment up here at the top, which is known as soluble APP alpha because it's based on the alpha secretase, APP alpha, and then we have this intracellular transmembrane sequence that we have down here, which is known as C83 peptide. Okay, so we get two fragments after alpha secretase cuts it. Pretty clear, I'm hoping. So if that continues, this C83 peptide is further cleaved by gamma secretase to produce what is known as P3, this other fragment. So imagine it's just this bottom part now that is being cleaved by gamma secretase. Gamma secretase will find its unique amino acid sequence and cut it right here. And as a result, it will produce this P3 segment that you see in the middle, and it will produce what is known as AICD, or the intracellular A-beta domain, or the A-beta intracellular domain. All of these different fragments, including APP and all of these other ones, are degraded and therefore do not, as far as we can determine, produce or possess neurotoxic properties. So this pathway, in other words, is safe. We want our amyloid precursor protein to be processed in this way. Yes? So all of them. So APP, P83, the AICD, all of these particular fragments based on this alpha gamma secretase, all of them are soluble and all of them are easily degraded. Okay? In particular, we're going to be paying attention to these fragments down here in a moment in the amyloid pathway. So the amyloid pathway actually is cleaved by a different secretase, hence its name. The beta secretase that makes this incision here will produce what is known as APP beta, and it will also produce what is known as the C99. So this is different. It's a different size. It's slightly longer. The amino acid sequences are also different, and it's about 16 amino acids longer than the C83, which is why it's the C99 peptide. This is characteristic of the the beta secretase pathway. So the C99, why does this become an issue? The C99 is further cleaved by this gamma secretase. It's the same gamma secretase, by the way. There's no difference. The gamma secretase in this pathway is the same as in this pathway, but the starting material that it's working with is actually different. So one of the things that results is that instead of forming this P3, we actually form what is known as an A-beta peptide. So this A beta peptide that you see in the middle here, this A beta peptide is the one that we think is neurotoxic. In experiments that were initially done at the TANS Neuroscience building where I was doing my graduate work, it was very clear and was shown very early on by a number of biochemists and researchers that the A beta peptide, if you applied it to neurons, is actually toxic. It will kill neurons if it is placed on them. And in particular, if you have what is known as A beta 40, and you will see that in the literature, if you have an A beta 40 or an A beta 42, those are particularly toxic to neurons. And so we know that this amyloid pathway is toxic to neurons in general. And these are the ones that we believe are going to be forming these different types of plaques, which will eventually kill these neurons. These are sitting outside the neurons. Remember, these are found on the outside. These things eventually get secreted out, and we find amyloid beta plaques outside of the neurons. We're not exactly sure what they're doing yet. There are a lot of theories and hypotheses, which you'll hear about in your fourth year courses, whether you're in neuroscience, cell biology, or if you go on to physiology, you'll hear more about these different processes. So I'm also going to mention, because there's some controversy about this, you won't be asked any questions about these neurofibrillary tangles, but in general, when we're looking at these two different theories, the other dominant theory, what happens in Alzheimer's, one of the dominant theories is we get amyloid beta. I just went through how you produce amyloid beta. We're going to come back and visit that in a few moments. The other dominant theory is somehow these neurons that are targeted within the medial temporal lobe first, and then they spread from your medial temporal lobes outwards. We think that something goes wrong inside the neurons, and somehow they start to produce these neurofibrillary tangles inside the neurons first, and whatever happens after is sort of just a consequence of this initial event of the generation of these neurofibrillary tangles. One of the constituent proteins that is a hallmark, so whenever you see neurofibrillary tangles, you should automatically be associating this with hyperphosphorylated tau. And this hyperphosphorylated tau seems to be the marker that we see within the degenerating Alzheimer's brain. So normally we have tau, just like we have APP. Our neurons have lots of tau because our neurons have lots of microtubules. When you have microtubules, which means that you have tubulin, when you have tubulin, in order to make tubulin stable, you have this protein known as tau to actually act to stabilize these microtubule containing tubulin polymers. And this is a really important consideration. We have all of these different things within the normal healthy neuron. We don't understand what causes it to go wrong in the first place. However, one of the things that we think happens, and there's some experimental proof that this is an early event, that somehow when it gets phosphorylated by a kinase, this tau loses its affinity for the microtubules and tubulin, and then it falls off of tubulin and it starts to congregate or to collectively gather with itself. And as you start to have less and less interactions between tau and more and more association of tau with itself, these microtubules start to disassemble. If microtubules are normally found within these long axons and they keep the axons functioning normally, and now the microtubules within these axons start to disintegrate, then you're going to start to lose normal neuronal function. And again, this is probably important for the normal axonal delivery. If you've lost the railroad between the cell body and the processes, the vesicles at these presynaptic terminals, if you've lost that railroad to deliver all these different proteins to the terminals at the end, then you're going to also be affecting neuronal survival as well as neuronal integrity. And that's the current theory. And we know that as we start to get more and more phosphorylation by these kinases of tau, that we start to be able to detect more of these phospho-tau proteins that form these large aggregates inside of the cell. Diagrammatically, I can show you that here, if these are the different microtubules, and you would have to assume that these are forming these different arrays all down the axon, that these are stabilized by these tau proteins that you see here. So this would be tau. Kinases come along and cause the phosphorylation of these tau proteins. And as they get phosphorylated, they no longer interact with tubulin. They will start to become hyperphosphorylated, and as they become hyperphosphorylated, they will now start to aggregate with each other, which is what you're seeing here, and eventually these will accumulate within the different neurons that are being affected by this process. Now, whether this occurs first and then other things happen, or whether beta amyloid occurs first and causes this to happen, we're totally unsure. There is no definitive proof either way that can show one group or the other that they have all of the correct sequence of data to account for all of these different phenomena. In either case, we know that in an Alzheimer's brain, we see all of this. We see beta amyloid, and we see hyperphosphorylated tau, but we just don't know which one occurs first, which one causes the disorder, and which one is a consequence of the disorder. You can see why it might be important because it is affecting a normal physiological protein that you normally have expressed. Now again, I'm not going to go through this in a lot of detail. You will see this in other courses, which is why I'm kind of bringing it up for you, and you don't need to worry about this for your final exam, at least in this course. So when we're looking at creating these Alzheimer's disease mouse models, there are so many of them. And in fact, here in Toronto, we've developed our very own transgenic animal model, and it's called the CRND mouse because the group that founded it was actually part of the Center for Research in Neurodegenerative Diseases, and the CRND mouse actually makes use of a lot of these different findings. So we can overexpress APP, we can affect what is known as presenilin-1. This will make some sense in a few moments, and this is one of the mutations that was found here at the University of Toronto, that when you have Alzheimer's disease that is inheritable, that is genetically based, and again, it's a small minority of that's actually genetic, that it involves presenilin-1 or presenilin-2, and we'll talk about this in a few moments, as well as tau. So again, if we're taking a look, we need to be able to, in all of these different mouse models, and if you're going on and you're taking other courses in neuroscience, or you're taking other courses in physiology or in cell molecular biology, one of the things that you have to be able to demonstrate in all of these different mice models is that it resembles the human disease. We should be able to, in a mouse model, be able to detect A beta plaques. Not all mouse models that are Alzheimer's, like models, will have beta amyloid plaques. We need to be able to demonstrate that. Not all mouse models, for example, will demonstrate these neurofibrillary tangles. We see them in humans, in human brains with Alzheimer's. We see both of these, but for example, the APP23 mouse that you see here that has A beta plaques actually don't have tangles. And it's the same thing for the PR5 mouse, that even though we see these neurons that you have here in black, that these neurons have neurofibrillary tangles, we actually don't see very many A beta plaques. But the human brain, we would actually see both. And that becomes, again, a really big issue. And again, that's for your own information to think about if you're going on to other courses. I do want you to think a little bit about this, though. And it might be one of those questions that you need to think about on the fly on your final exam, although I'm going to give you a little bit of a heads up to think about that along the way. So if it's A beta, and we're worried about developing these A beta plaques, somehow we need to either stimulate alpha secretase, or we need to somehow inhibit some of those gamma secretases. We might want to inhibit the beta secretase. We might be thinking, as many individuals have, and Joanne McLaurin, who's in LMP here at the University of Toronto, is actually researching whether or not we can fix Alzheimer's disease by doing either passive immunization, where you're giving antibodies against the A beta plaque, trying to destroy the existing A beta plaques, or to use active immunization, and you yourself will develop these A beta peptides. But so far, the research hasn't been very compelling. There isn't a good immune response that will clear your brain or the brain of anyone who has Alzheimer's with these different strategies that involve a knowledge of immunology. So somehow, if we're looking at this other possibility where it involves these neurofibrillary tangles, we either have to inhibit that initial phosphorylation, we have to inhibit the phosphokinase, in other words, to inhibit this tau aggregation, or we could do it the other way. We might stimulate a phosphatase. If we can eliminate the kinase from working, or we can block it from working, great. But if we can't, maybe we can actually remove those phosphate groups so that we don't have hyperphosphorylated tau that might lead to its aggregation. There might be other ways of increasing microtubule stabilization. So there might be other drugs that could be used to stimulate and stabilize these microtubules. And again, there's another possibility of increasing tau clearance. Can you see the potential problem with this last therapeutic approach? Because all of these are in the literature. What's the problem with trying to increase tau clearance? Where is tau normally found? Yeah? In the axon. So normally in the cell, like how are you going to try to clear it from inside the cell if it's actually a really important normal physiological player in all of this? So again, the statistics and some of the numbers that are up here not being really important, but I wanted to talk to you about some of the genetics of Alzheimer's disease. You don't have to worry about any of this. You only have 2 multiple choice test questions. I can guarantee you the multiple choice don't deal with any of these numbers, and I'll tell you when you should be paying attention again, but I do want you to have a foundational knowledge of Alzheimer's as you leave this course. So sporadic Alzheimer's disease, or sporadic AD, where we can't account, like what causes this, actually accounts for the majority of Alzheimer's disease patients. We don't know the cause. You go in, you get a neurologist who diagnoses you with, you might have Alzheimer's, you might have this, you might have that, but you probably have Alzheimer's. That's a sporadic form. The familial forms of Alzheimer's disease only account for about 5%. It's the minority. We don't know what causes this. Familial forms of Alzheimer's disease generally appear much earlier, in the late 40s into your early 50s. Those are really bad forms of Alzheimer's disease as well. And I say bad in the sense that it's very, very rapid, very progressive. They lead to rapid personality changes, loss of motor sensory function. All of those get affected in these familial forms. The symptoms overall, though, we still have memory loss. We still have all the other symptoms that we have looked at before. Even though it's only 5%, about 50% of all of the genetics. So someone in your family has this, someone else in your family has Alzheimer's disease, your uncle had it, your grandfather had it, et cetera, and you're doing a normal family history, 50% of familial Alzheimer's disease can actually be explained by either changes in genes encoding APP, which makes sense. There's a change in amyloid precursor protein, which makes it more likely that you'll form the A beta form rather than the alpha form, or these proteins that I told you were found here at U of T, which are known as presenilin 1 and presenilin 2. This was the work of Dr. Peter St. George Hislop, whose lab was on the first floor of the TANS Neuroscience building, who is just down the hall from us. APP mutations, again, you don't need to worry about this slide, are relatively rare. Only about 12% of all familial Alzheimer's disease patients have APP mutations. Average onset fairly early in the 50s. The people started to investigate these APP mutations because Down syndrome individuals actually develop Alzheimer's disease after the age of 40. So again, Down syndrome is a genetic disorder that has a lot of common links, and it looks in some ways a lot like Alzheimer's disease. And in those individuals after the age of 40, most of them will actually develop Alzheimer's disease after the age of 40. So it's sped up even more in the genetic background of Down syndrome. One of the reasons why they thought it was genetic, and they thought it might be due to APP. APP happens to sit on chromosome 21. You have 3 copies in Down syndrome of chromosome 21, so this might be the smoking gun that people were looking for, and indeed, when they looked, when you have extra copies on chromosome 21, you actually increase the likelihood of having these mutations. So again, there's increased cleavage. It might be due to an increase in the beta secretase or making it more likely or more amenable to beta secretase, and you're going to get more of the A beta 42 and A beta 40. And these are ones that are particularly prone to be forming those different plaques. I also mentioned that of the 50%, of the familial forms of Alzheimer's is this protein here, presenilin. And again, I'm not going to differentiate between presenilin 1 and 2. I do need you to know presenilins, and I do need you to know different things about presenilins on this particular diagram. So I told you earlier that gamma secretase is one of those enzymes that seems to be really important. We can have gamma secretase in both the alpha pathway and the beta pathway. But this gamma secretase actually has 4 different constituents that I'd like you to make sure that you are aware of. One of these is presenilin 1 or presenilin 2. They can have either of those. That's component 1. They can also have another component which is known as nicastrin. They can have APH1, and then they can have this other molecule known as PEN2. Now again, I don't need you to worry about all of the details here. The reason why we talk about gamma secretase, and I told you that it is going to cut the APP or the fragment C99 or C83. The presenilin is the only component of the gamma secretase. Out of all of these other ones, presenilin is the only one that actually causes proteins to be cut or enzymatically digested. It is the only one with proteolytic activity. So again, mutations in presenilin proteins actually make it more likely to develop Alzheimer's disease at an earlier age. And again, these presenilin mutations increase the likelihood and increase the overall activity of the gamma secretase that we're examining. So I'm just going to go through this one series of slides, then we'll take a break. I promise it'll be relatively short today in terms of the lecture. So even though I'm a little over time, I have to apologize. We'll make sure that you get everything that you need for the final exam, and we'll still have an in-class review. So the genetics actually explain only a very small proportion, 5%. Remember, the vast majority, Alzheimer's detected familial forms, only about 5%. Although we keep looking, and although we have a lot of genetic capacity now, so we're able to do whole exome sequencing, whole genome sequencing, it's incredibly fast. Our ability to annotate using different programs allows us to screen someone's genetics within about a week, which is much, much faster than it used to be a long time ago. And although we keep doing this, we haven't identified genes that we can say with 100% certainty that this is what is responsible for Alzheimer's. Again, the vast majority seems to be sporadic for reasons that we are not able to currently describe. However, there are a few that are very clear in the literature of increasing what is known as the relative risk or the odds ratio. If you have this, the odds of you developing Alzheimer's is going to be 3 times higher or 2.5 times higher than you would if you don't have this. One of those risk factors is known as apolipoprotein E, often abbreviated as ApoE. Many of you who have heard about Alzheimer's will have also heard about ApoE. In particular, there is one variant of ApoE, ApoE4, but I want you to understand that, and I want you to understand why ApoE4 actually might be a big risk predictor for late onset, after the age of 65, forms of Alzheimer's disease. So apolipoprotein E comes in a number of different isoforms. So we have ApoE2, ApoE3, and ApoE4. So ApoE is normally produced by the microglia. So again, think about glial cells in this last lecture. Think about the importance of these microglial cells. They're the normal immune cells of the brain, but these are also carrying an important role because ApoE is produced by the microglial cells as well as some astrocytes within the brain. So apolipoprotein E is normally important because it surrounds and solubilizes cholesterol. So especially within the brain, because we have lots of different reasons why we want cholesterol in the brain, we need to be able to transport it well, and we need to be able to contain it well. So these three different alleles for ApoE have been identified: ApoE2, E3, as well as E4. The vast majority of you sitting here in this room actually have 2 copies of ApoE3. I can say that with 100% certainty based on statistics that probably about 80% to 85% of you have 2 copies of ApoE3. It's been found that a small number will actually have ApoE4. And ApoE4 confers what is known as the risk of developing Alzheimer's. It does not mean that if you have 2 copies of ApoE4, it's game over, I'm going to develop Alzheimer's disease. That's not what it means. But it does mean that you increase the risk of developing Alzheimer's disease if you have 2 copies of ApoE4. This puts you at an increased risk, where we have a switch in one of the amino acids from cysteine into an arginine. If you are one of those really, really rare individuals that have ApoE2, and you have 2 copies of ApoE2, you're actually protected from developing Alzheimer's disease. Doesn't that sound great? Would you want to have ApoE2? No? You wouldn't. Okay, why not?
There are probably disadvantages.
There are probably disadvantages, and you're 100% correct. So even though ApoE2 is protective against Alzheimer's disease, it actually puts you at increased risk of developing coronary heart disease. So you either have Alzheimer's or you die of a heart attack. So the best thing is actually just to be normal. Be normal. Being normal is okay. So having 2 copies of ApoE3 is actually the best strategy overall. Or having 1 copy of ApoE3 and 1 of ApoE2. So why is all of this important? And then we'll take a break. You do have to know this. You're going to see this on your final exam, and I don't want it to be a surprise to you. You're still going to have to study it yourself. You're going to have to think about it, but you're going to see this in parts of this slide on your final exam. So why ApoE? Why do we care? What's its relationship to this A beta? Why do we keep hearing about ApoE4 and the risk factors for Alzheimer's disease? So ApoE has been shown to actually bind to and clear from the brain A beta. And it's been found to increase the clearance of A beta from the brain. If we start decreasing the levels of ApoE, it actually means that we increase the amyloid deposition. The form that seems to bind the least with amyloid beta. So if we're producing amyloid beta, and this is a mechanism, if once we produce it, to eliminate it, ApoE4 actually is the isoform that correlates with the least or the lowest amount of A beta. So if you have rolled the dice and you happen to be really unlucky and you have overactivity of one of the gamma secretases because you have a presenilin mutation, and you also have ApoE4, then not only are you producing more A beta, but you're also eliminating it from the brain less and less. Another important consideration is this lipidation status. You have to lipidate or put a lipid group on ApoE to increase its activity, which also helps in the A beta clearance. What causes this lipidation is this protein known as ABC1 or ABCA1, which is really important, and we'll talk about this and the next diagram after the break. So I think now is actually a great time to take a break. If you have any things that you need to think about, now's the time to sort of think a little bit more about them. So a lot of people actually think that all of these mechanisms actually look like trying to regress back, but it's stuck in the chain. Obviously, try to go back in the cell cycle, back to where it was originally in development. That's a theory too. Okay. No, you're going. Are they quick questions? They're not quick questions? It doesn't. No, we mutate some of the GFP amino acids, sequential fluorescence, and then to activate fluorescence in the large There's
What's the purpose of light?
suffering. According to Buddha, it's suffering.
Two bars, I guess.
Okay, if you don't mind, I'm going to try to get going. So this is actually a really important slide for you to make sure that you go back and review. And this review actually is important because a short answer question revolves around you understanding this particular pathway. So we were looking at the relationship between ApoE. Some ApoE binds A beta better, but we're just looking at ApoE in general. So ApoE4 would do this the least, and ApoE2 would do this the best, but all ApoEs bind and help in A beta clearance. So this would be the general mechanism. ApoE4 wouldn't do this so well, it wouldn't happen as often, but this is sort of the general mechanism by which ApoE helps in the clearance and facilitation of its degradation of the A beta peptide. So we know that we have this A beta, so you should know like the different steps that are involved in taking amyloid precursor protein, attacking it with beta secretase, then having gamma secretase coming in, taking that C99 protein that results and then cleaving it into both the amyloid beta protein and the AICD that I mentioned to you earlier, and ultimately it may produce something like this soluble A beta. In certain situations, we know that it can be facilitated, the degradation of this soluble A beta that's sitting out here can be facilitated by another enzyme which you may not suspect might be that important. We have as this other enzyme, insulin degrading enzyme. And again, for those of you who are in nutrition or thinking about other physiology courses, your brain uses glucose in a very different way. It actually does not require insulin to have glucose being taken up by neurons. So we don't need that. We don't need like a glucose or a GLUT4 molecule, etc. But we do know that insulin degrading enzyme exists at really high levels throughout the brain. And one of the reasons why we think that this enzyme that normally might break down brain insulin, and again, that's a story in and of itself, this insulin degrading enzyme will actually help to break down A beta even further. This is helped, as you see down here, by having lipidated ApoE. So this breakdown is further enhanced by having ApoE that it is going to help in eliminating or breaking down the A beta. So the other thing that ApoE will do is this lipidated form of ApoE will help in the pinocytosis or the uptake of the soluble A beta, through binding to niprolysin, or NEP. It will take it up into these microglial cells, the immune scavenger system within the brain. And inside the cell, this complex will be further broken down by the microglial lysosomal system that you see here. So this lipidated ApoE functions in two ways. One, to enhance the activity of insulin-degrading enzyme, and two, to enhance the internalization of the A-beta into these immune cells like the microglial cells. One other thing that you might want to think a little bit about: how do we actually form these different ApoEs? What actually produces ApoE within your brain? So without getting into all of the details that you don't need to worry about, the enhancer elements that are found or the promoter regions that are found, within the astrocytes and microglial cells, we actually have a number of sequences like LXR. Those are sequences that normally will bind to different DNA elements, or RXR, or PPAR receptors, or PPAR. These become activated, and one of the things that these will do is to increase that ABCA1 molecule that we talked about earlier. So it will increase the production of ABCA1 as well as increasing the amount of ApoE overall. So why, again, is all of that important? So ApoE is important because it will bind to and become lipidated, and this process, after it becomes lipidated, is further enhanced by this process. So if you can increase the levels of ApoE by activating these LXR, RXR motifs within these glial cells like astrocytes and microglia, that would increase the levels of ApoE. We can further lipidate the ApoE, and that will help in both of these processes that you see here on the left, on the bottom of this particular diagram. And again, that will further enhance the degradation by insulin-degrading enzyme, etc. And this is what we want. Overall, we want to decrease the amyloid beta plaque load. And if we can somehow do this, which is what the strategy has been, to find drugs that will actually drive this process to increase the levels of ApoE, especially if you have normal ApoE3 status, then this might be somewhat beneficial. So the problem with all of these things are people have been spending a lot of time and effort on this. And in fact, even last year, which was really bad news for neuroscience and neurobiology, that overall, when scientists have taken things all the way through almost to full use in the marketplace, that when they got to these phase 3 trials, these drugs failed in those individuals. So people had spent like $1 billion to $2 billion doing animal studies, then doing early clinical trials, and from these clinical trials, just using further phase 3 trials. And at the end of these phase 3 trials, these drugs had so many serious side effects in humans that these drugs were actually abandoned. And that happened last year for 2 different Alzheimer-related drugs. And so people are always looking at these different therapeutic interventions, but it becomes really problematic. We know that a lot of these therapies work incredibly well, just like they do in stroke. And I told you that in stroke, a calcium blocker will prevent stroke 100% in animals, especially in mice, but they don't work in humans. It's the same thing that we're finding in terms of clinical trials involving Alzheimer's disease therapeutics. Again, plaque clearance in Alzheimer's can, in some cases, be achieved, but the cognitive decline can't be stopped or reversed, except for one study. One study has shown that if you block plaques from forming and then eliminate plaques after they've been formed, you can actually reverse everything: memory decline, personality changes, etc. But those studies actually haven't been replicated, and we're still kind of waiting for other people to do this. We don't have all the answers. Some of those answers I'm going to have to leave to you to find, because these are answers that some of the best minds in neuroscience have been trying for over 20 to 30 years. Sharing with one of your colleagues during the break that I know still to this day, a lot of these different Alzheimer's disease researchers, many of them here in Toronto. So some of you might work at UHN with people like Dr. Anurag Tandon, or you might work with Dr. St. George Hislop, still very active in research over at the UHN. Some of you might work with Paul Fraser and others, but they've been working on this problem for decades. Many labs have been working on this for decades, like Dennis Selko's lab and others, but we still have no solution for this. Some of the more interesting interventions actually don't come from the world of therapeutics. They actually come from the world of clinical studies. And this was actually really, really cool. This was only 2 years ago. It's been replicated, found to be incredibly efficient in human trials that have been tried so far. Some of you might have seen the news report coming out of Sunnybrook Hospital. Sunnybrook Hospital is actually the lead on these types of focused ultrasound studies. By breaking down the blood-brain barrier, we can deliver drugs directly to the brain, and we think that that's a better way to do it than to inject drugs into the brain or drill holes in the brain. A lot of drugs that you take orally can't pass through the blood-brain barrier, and we know that focused ultrasound and scanning ultrasound has actually been incredibly effective in removing these amyloid beta plaques, not only in mice. It's now been moved toward human studies as well, much of that being pioneered right here in Toronto, which again just shows you how much time and investment in neuroscience research U of T and the Toronto research community has undertaken. Do you have any questions about anything we've covered so far? Or yes, Raquel?
Quality of life for patients who undergo these types of treatments?
Pretty good.
So
It's
like they don't
it's an outpatient basis.
therapy, but okay.
Nope, it's pretty much an outpatient basis. There are a limited number of human clinical trials going on right now. There are few that are actually enrolled at Sunnybrook, but they are enrolling individuals with Alzheimer's disease at Sunnybrook. Okay, so now it's time to go through some of the other things that I want you to make sure that you know for your final exam. Before I get there, I just wanted to just tell you that no matter what happens on the final exam, because I hope all of you do brilliantly on the final exam, I'm going to try to make sure that you do brilliantly on the final exam. A lot of that's up to you, but I want to make sure that you all understand that you're really good students. An exam doesn't really say that you're a good student, bad student, etc. It just means that for that one exam, something might not have gone in your favor, but I would never think any less of you, so I want you to understand that sort of ahead of time. But with that said, I do expect you all to do really well on your final exam. Okay, so I told you that when I was here, Rija, this is my hair. You had asked pictures, hair, there it is. So when I was here in undergrad at U of T, actually, for the most part, I really didn't have a lot of interactions with my classmates, and I really showed up to class when I needed to show up to class, and most of my time was actually taking pictures like this, I guess. And it wasn't really a great experience. I did okay in some classes, but for the most part, I was pretty much a C or C plus average student. And for the most part, my life was sort of just going with the flow. I did what everyone else in my class did, and I did whatever anyone else like in my major did. If they took that course, I just ended up in the same course as them. And for, again, the 4 years that I was here in my undergrad, it went by really, really quickly. I wish, in retrospect, that I had sort of done things a lot differently, which is why I insist that you guys do things differently as undergrad. So I want you to have a better experience than I had when I was an undergrad, and I hope that's something that you keep with you wherever you go. Because believe it or not, even though you're in third year, and some of you might be slightly more advanced in terms of the number of courses that you've taken, all of you are going to eventually end up here in Con Hall. You're going to graduate sooner than you think. And my 4 years went by as a blur. Literally, it was like floating on these waves. I took courses, I failed exams, I passed some exams, I did some assignments, I failed some assignments, but ultimately I ended up graduating. But I have like no recollection of my undergrad experience other than I hated physical chemistry, and I kind of liked physiology lab courses, and some of my seminars were really cool, but to be honest, if you were to ask me, I couldn't tell you a lot about my undergrad experience other than one day I woke up and I went to Convocation Hall and I went and sat through the 2.5 hour ceremony, and it was a very unappetizing experience. So while I was here, my mom asked me, and again, so she's asking me here as we're walking off toward UC, what are you going to do next after your undergrad? And I told my mom, quite frankly, after finishing my undergrad, I have no idea. Like, I don't know what I'm going to be doing next. And it's one of those things that I had no plan to do whatever was supposed to come my way. I had no idea what was going to come my way. And as I've shared with some of you, my greatest hope in life after graduating from U of T was applying to the CIBC, TD Bank, Scotiabank to become a bank teller. That was like my greatest hope. Like, I really hope they take me. That was the only thing going through my mind. And I'm kind of glad that that wasn't something that actually came through. And I'll also tell you that even though things might not look all that great for you right now, ultimately everything does work out. I told you that even without a stellar undergrad experience, I got into graduate school. I overcompensated, to be quite honest. You can't tell from this picture, but I'm starting to lose my hair after doing my master's. Like, if you're spending 20 hours a day, every single day in the lab, it's going to take its toll on you. And I overcompensated by trying to be a better student by a lot in graduate school. But ultimately, what I learned from all of this was that even though I didn't have a great undergrad experience, and to be honest, I didn't have a great graduate school experience, that I learned a lot along the way. And that even though all the plans that I had in undergrad didn't turn out, that overall everything wasn't as bad as I thought it was going to be. I thought literally, for whatever reasons, I thought I would be living out on the street, kind of trying to make a living doing whatever, I'm not sure. But ultimately everything did work out. So I want you to have that same sort of attitude. So again, we sort of started looking at this at the very beginning, sort of as a journey, and now our journey is coming to an end. And so as we start going back and we start thinking about this journey that we've taken, I'm hoping that many of you will have remembered the movie Lord of the Rings, and apologies to my 3 colleagues there. I think you've seen this before. You can't remember. You kind of remember. Okay. So I don't know if you guys remember the movie Lord of the Rings at all. Kind of, yes. But for me, it was one of those really great movies because, you know, they're in this quest to destroy this one ring, and this was done sort of as a group, and now you're about to go and destroy that final exam, sort of that evil final exam. And of course, that one ring that we're looking at here, you can look at it in a slightly different way. Many of you are on a slightly different quest, not just to destroy the final exam, but also to get that magical 4.0, right? And some of you are kind of thinking, like, how do I complete this journey, get all the way to that magical land of the 4.0s? How do I make sure that I have those right rings in the right places? So again, I just wanted to share with you, no matter what happens on the final, even though I want all of you to succeed and I want to have those office hours for you in the next coming days, that I want you to remember that sometimes, even if you think that you can't do well on the final, please let me help you do that, because I don't want you to give up along the way. I don't know what kind of obstacles might be facing you right now, whether it's stuff at home, whether you're having trouble concentrating or whatever it is. I don't want you to doubt yourself. Often I did. I doubted myself an awful lot in undergrad. So I had switched out of my initial medical genetics program. I switched into Farm Talks. I didn't really like Farm Talks, so I switched into physiology. So I really doubted myself a lot along the way. But I was also really happy because I had some help along the way as well. So as we kind of get ready for the last part of our journey, as you get to visit that scary place on Wednesday evening at 7 o'clock, and you hope to do the impossible, maintain that U of T 4.0, I want you to make sure that you understand that there's always some help for you if you need it, especially over the next couple of days. Keep things in perspective. You have lots of time to be able to do all the things that you want to in this course. None of you are in serious trouble of failing this course, which is great. And many of you are in the position where you're going to do great. And so I want to make sure that you have the proper roadmap to get you to that destination, whatever it is, whether it's total and utter destruction of the final exam or maintaining that 4.0. I know you don't have these notes. I will post these for you a little bit later. Just a couple of things that I want you to keep in mind. So you have these 20 multiple choice test questions and these 4 short answers with parts. All of you are going to do question 21, and then you have a choice out of the 3 of the remaining 4, as I mentioned to you before. Each of those short answer questions is worth 10 marks each. Most of them have either 2 or 3 parts that are associated with them, so one question by itself won't make or break you on the short answer. So you're going to have a full 2-hour final exam. I'm hoping that most of you will have more than enough time to check over your answers, and I hope that you will go over your answers, not just finish and then leave. I want you to make sure that you go over the online quizzes. I'm not sure if you guys still have access to all the online quizzes, do you? If you don't, then come and see me, or I'll post the questions from the online quizzes so that you can go back and review them. I promise you that they will be helpful. Like, I don't want to just give you quizzes for the sake of giving you quizzes. They will be very helpful for you on the final exam, especially on the multiple choice. Use those 2 review sheets that I provided for you. They'll give you a lot of help and some guidance. You can ask me anything during the next 30 minutes or so related to these different topics. I'm not going to answer individual questions of, do we have to know this during the next couple of days? Because I want everyone to have equal access to me. So today's the day that you can ask me anything, and I'll answer for the entire group. If there are any questions from online, you can let me know as well. Okay, so again, I want you, here's an important consideration, because I know that this is going to be something that you need to think about. In general, we've been talking about excitatory synapses, and therefore we've been talking about the postsynaptic potentials that arise from those. And we've abbreviated these as EPSPs. If you have already started to review lecture 7, then you'll have read through synaptic plasticity. These same rules also apply, even though we haven't talked about them, to inhibitory synapses as well, especially the short-term forms of plasticity. So if you're thinking about short-term plasticity in this first bullet point, that includes things like facilitation. Can I facilitate an inhibitory synapse? Yes, I can. Can I augment an inhibitory synapse? I can. Can I have short-term depression on that synapse? I can. And again, I want you to make sure that you understand that. It is part of your plasticity notes. It's one of those bullet points. But I don't want there to be any surprises for you. That's like the one thing that I've learned, that sometimes one surprise can actually lead to a cascade effect. So I want you to understand that ahead of time. So if you're looking at an IPSP or an EPSP, we can talk about the same mechanisms overall. You should definitely make sure that you know the different blockers and the drugs that we've talked about in class. It will be very helpful for you on your final exam, especially if you're going to answer one of the short answer questions. It's useful to make sure that you know that. I also want you to be aware that short bursts of activity or stimulating presynaptic neurons, etc., versus like 100 hertz or 100 action potentials or 1 action potential per second, those are things that you can use abbreviations for, but I'm also just reminding you of what these different things are. I'm going to mention also that Dale's hypothesis shows up just once on your final exam. It's not the focus of the question, so you might be thinking, what does he mean by that? But you'll understand that when that time comes around. So Dale's hypothesis, know what it means, but it's actually not the focus of the question at all. So do you have any questions just generally before I go through lecture by lecture? Any general question format questions? Any questions? Yes, Raquel?
I guess it could also be in lecture 7, but about post-tetanic potentiation.
Yep.
Like, you wouldn't see that experimentally, right? Because tetanus is not a physiological phenomenon.
The reality is, would you see post-tetanic potentiation? We see it all the time in long-term potentiation experiments, because that's what we see at the beginning, and then it kind of slopes back down. that part that's stable, that's what we call LTP, but that increase that we observe in the first few minutes is actually the post-tetanic potentiation. Okay, so if you don't have any other questions, or are there any questions from people online? Nope. Are there any people online?
Yes.
Okay, 6. All right. So for lecture 5, and particularly I want you to make sure that when you're going through lecture 5, you know a little bit about the presynaptic machinery as well as about KD. So what do you equate with KD when you see KD? So whoever's wrong. So Carl Diceroth, right? So neurobiologist, KD equals Carl Diceroth. If you're watching an NBA game, Kevin Durant, sure. If you're eating dinner, Kraft dinner is fine, but we're thinking Carl Diceroth. I want you to make sure that you know the optogenetics section. So I would suggest very strongly that you know the details and the proteins that are involved presynaptically for neurotransmitter release. In particular, I'd like you to make sure that you know about synaptotagmins and what the C2, C3 domains are. I promise you the multiple choice, like if you know this, it'll be fine. I'd like you to make sure that you know the different protein markers that therefore represent the different genes that would allow you to target specific elements to specific groups of neurons. I'd like you to also make sure, if this is news to you or you've never heard this phrase before, some of you who have studied, are you thinking intrinsic fluorescence? Do you know what that means? So monoamines, which means nothing. What does that mean? Monoamines have intrinsic fluorescence. So when you shine UV light on them, that they will fluoresce automatically. I don't have to stain them, so go back and revisit that a little bit. So you should also know, and this again, you'll get 0 on this question if you answer it incorrectly. So when I'm asking you to help visualize a gene, if you are putting down as part of your answer that you're putting together as a genetic construct, if you are putting down something other than a gene like mCherry is a genetic sequence, GFP is a genetic sequence, DS-RED or DS-RED2 is a genetic sequence that I can put into DNA. I cannot put things like DAPI. DAPI is not a genetic sequence. Okay? So I want you to be very clear on that. I can't emphasize that enough. That question, if you were to put, like, I will have channel rhodopsin plus, like, DAPI, you would get 0. Even though channel rhodopsin might be correct, I can't tell if you understand what DNA is. I can't tell if you understand how to package things into a virus. If I'm asking you, and you see this keyword visualize, it means put in a gene that gets expressed with another gene like channel rhodopsin, like mCherry or GFP or DS-RED or enhanced yellow fluorescent protein, EYFP, whatever it is. So make sure that you understand that. So again, from lecture 5, you get 6 multiple choice test questions. One of those involves proteins of the presynaptic machinery that allows you to release, as well as the CAZ. What is the CAZ? So the cytomatrix active zone, so again, a presynaptic designation. Again, all of you will answer question 21, which is a pure molecular question to make sure that you understand how to draw a construct, how to go back and verify a construct, tell me about different viruses, etc. Andy? Yep. Yes. Yep. Yep. Do you have another question?
Yes. Remember, like for just looking at constructs, like double colons, like what does that mean?
So are you talking about like TH double colon IRES CRE? You don't need to worry about that. It's a designation that we have to delineate between like an internal ribosome entry site and the gene promoter region. Like you don't have to worry. Okay? Any other questions? Okay, so again, make sure that you can do all of that. For question 21, I want you to read very, very carefully. Some of the answers that you've been putting online are good, but I want you to read carefully. There is a difference between asking you for activation. So for example, I can turn on a group of neurons using channel rhodopsin 2 or CHR2, I can activate them, and that's different than excitatory neurons. So in general, when I'm talking excitatory neurons, you should automatically equate excitatory neurons with the glutamatergic system. Here's my rationale for why. Some of you argue cogently, coherently, that many neurotransmitters, like dopamine, for example, could be excitatory. Absolutely correct. But dopamine can also be inhibitory. There's a lot of ambiguity. When I say excitatory and it's glutamate, glutamate is always excitatory. It doesn't matter the system that you're looking at. So if you see excitatory neurons, should equate that automatically with the glutamatergic system. For question 21, you are going to be asked to draw something related to many of the scenarios that I've given you on that worksheet. So therefore, you should make sure that you're comfortable with drawing on that worksheet that I've given you earlier. You should know what the importance of Cree driver lines are. And you should know what genes can be used for visualization. Again, DAPI is not a gene. It is a nuclear stain that helps us to see nuclei. But if you were to put that as a genetic sequence, it would be completely wrong. You would get 0 on that particular question. You had another question. Yes?
So for instance, when you're drawing a construct, for instance, if you're targeting glutamatergic neurons, one of the possible protein markers is VGLU2.
Yep.
If you want to use a promoter for it, you always have to put a P next to it.
As long as you somehow have indicated that you were using a promoter for a specific system, whether it's a P or some other, you can spell out the whole word, it's up to you.
So do you have to put PVGLU2 or just put VGLU2 as GLU2?
I have no idea what VGLU2 would mean.
As a promoter region, you have to put down promoter, right? It's ITR.
ITR is not a promoter region. It's an
ITR, then you get the promoter, then lost P, whatever. In the promoter region, you have to put P next to the specific.
I would say it's always better to make sure that there's no ambiguity in your responses. And if you're writing out promoter, fine. But if you're just writing things down and we can't tell if you understand how the system works, then it's not something that you're going to get full marks for. Okay, so just try to eliminate any ambiguity. If you want to write P, fine. We understand that P stands for promoter. If you want to write out promoter, also fine. You can do that as well. Again, you have a few days to drop by the office. I still have a little bit of time tomorrow afternoon after about 3 o'clock. If you want to drop in and just go over some of the things that you've written out, I'd be happy to do so. Again, just a little bit of a hint for you. You will be given a scenario where you have to go back and figure out, based on what you know, and we've gone through this, because I think it's a really important part of your training as a molecular biologist, or in cell and systems biology, or as a neurophysiologist. More and more times you're now asked to read papers that involve viral delivery. Based on the scenario that you're given, what kind of virus was likely used, or why is that virus being used for this type of construct? You should know some of the major features, the sizes, the types of genetic information that's put into it, the speed of expression, how long it lasts, etc. So, good question. I've already indicated that for you before. Make sure that you know that. So, question 21 is the only short answer question where you're going to be asked to specifically about a genetic circuit. Again, excitatory generally means glutamate. Inhibitory generally means GABAergic. So use these if there's no other instructions given to you. So I could, for example, argue that 5-HT is inhibitory. Absolutely true. In some neurons, serotonin is inhibitory, but in other neurons, serotonin is inhibitory. GABA is always inhibitory. It doesn't matter where you're looking. So again, inhibitory, I would want that, I would strongly recommend using GABA. And again, think a little bit about the different viruses and packaging. It will be really useful for a short answer. You should think about DREADs as well. You should know like what sort of keywords are associated with DREADs that would allow you to make sure that you're using DREADs versus like using optogenetics. Okay. So are there any questions online? No. Okay. So I'm going to tell you that you should make sure that you know the structure of the hippocampus. There is a multiple choice test question where you're told something happens to a specific neuronal group. You have to deduce which neuronal group or axons is being stimulated to cause that result. You don't have to worry about differentiating because a lot of you have been really panicking about this concept, about the differences between associative and non-associative LTP. You don't have to worry about that. So again, I want to reduce your stress levels as much as possible. You're not going to be asked any questions about differentiating between associative, non-associative LTP. So please don't stress over that. Again, make sure that you've understood all of the drugs that are used to block the different types of receptors. Should know the structure of the glutamate receptors, and make sure that you know the parts of this section. And there are 2 short answers actually based on what is covered in this entire lecture. So this includes the structure function of the glutamate receptors as well as about knowing the differences between early and late phase glutamate responses. Okay? Do you have any questions on this or anything that you're not sure about what I'm talking about or what I'm indicating here? Raquel, yes? Another question?
Glutamate response curve, like do we have to know exactly like which millivolts, like the early phase disappears and
I'd say it's a pretty good idea to make sure that you know the different voltages that you might use in voltage clamp experiments. Okay, so I'm going to also mention this because, again, I don't want there to be surprises. I want all of you to do as well as you prepared for this final exam. I want it to be symmetrical with how much preparation you put in. So if you know all the material, you should be able to get 100. I don't want it to be a surprise or a shock, and I want it to be a fair exam. So you should know about spike timing dependent plasticity. You should understand how it could be induced. What could I do to make STDP, whether it's negative or whether it's a positive spike timing interval? Positive spike timing intervals, where I stimulate presynaptically first and depolarize after, always creates strengthening if the timing is short enough. And negative spike timing intervals on the left side of the diagram always means that you get a weaker synapse that looks like LTD. Make sure you understand how that works. And whether or not this is physiological. You should make sure that you know the superecliptic Florin system, how you would use it, how you might apply it. And again, some other things for you to think about. Is the kainate acid or kainic acid receptor subunit the same as the NMDA receptor subunit in terms of its topology, in terms of how it forms a receptor? Is it the same or not? Overall, if I were to draw it, would you be able to tell the difference between an NMDA receptor subunit and a kainate acid receptor subunit? Or would they look pretty much the same? Pretty much the same, correct. Do they also form do they need 4 receptor subunits or do they need 5 receptor subunits? So they require 4. So again, I want you to make sure that you are aware of that. It's not just AMPA, all of these are the same. So you should also be thinking about global synaptic scaling. What is it? How do I use different substances to induce scaling? So what happens when I have potassium chloride in high concentrations? What happens if I have TTX in high concentrations, etc.? And I want you to also think about this, because I've mentioned it, but it's not a clear concept for about a third of you, I would say, wouldn't be able to answer this correctly right now. So I want to completely clear that up now. If I am looking at the mechanisms that I use to induce LTP, you guys know about high frequency stimulation, tetanic stimulation. If I am looking at the same mechanism, I should be able to see the same thing with spike timing dependent plasticity. So STDP, I would expect exactly the same thing. I would expect AMPA receptors, an increase in CAM kinase 2, etc. So don't worry about the long-term forms. They're always identical. So if you see spike timing dependent, you use the same mechanism that you know for LTP, CAM kinase 2, NMDA receptor activation. You can block NMDA receptors. But short-term forms are always different, right? Do you know the differences in short-term forms of plasticity? Those are always presynaptic or generally presynaptic. They involve changes in calcium, right? And those are different. So anytime you see a long-term change, that is something that you should equate with LTP, the mechanism that you know in detail. STDP, any of the other ones that we've covered in class. So for lecture 8 for stroke, again, I would ask that you make sure that you know the differences between early and late glutamate responses, and think a little bit about this in advance. So how would they change the early versus late? How would they change for LTP? And would they change in the same way? And yes, they would. So I'll just tell you that. For STDP, you would see the same changes for LTP and STDP. Okay, so which of those are going to change? In other words, is it AMPA that's changing or NMDA that's changing? LTP, we change. Are you going to disappoint me on Wednesday evening? It's AMPA, right? AMPA changes, NMDA never changes. But again, you're going to be asked that in a slightly different way, but you need to think about it, and I don't want there to be any surprises there for you. I'm going to tell you in advance that you are required to know how we induce stroke in different animal models. So I went through about 5 or 6. You don't need to know and memorize all 5 or 6, but you should be able to tell me about most of those. Again, you'll see this both on short answer, and you'll be able to relate this back on like a multiple choice test question. You're going to see these in other fourth year courses, so I want you to make sure that you know those. It's very rare that I ask you to know specific results from a paper, but from that paper on axonal sprouting, I want you to know the function of what ATRX is, and I also want you to know the overall expression pattern changes for LINGO and the NOGO that was presented for you in that paper as well. Again, I don't want there to be any surprises for you. You're still going to have to do the studying. It's still going to be challenging enough, but I don't want you to be overly stressed, and I want you to start working together to think about your final exam. Okay. Here is a good example of a question that you might be asked related to stroke. So if a researcher creates an animal model of stroke, how would they assess brain damage or damage to the brain using a staining technique for cell death? What is that really asking you? So what marker could you use for cell death? What marker do you know of right now off the top of your head? Sorry? Collar toxin B in this case would not be the answer I'm looking for. Corey? Could use caspase 3. Do you remember that mitochondrial protein? Cytochrome C. Could use those. Those are markers for cell death in neurons. And then what about immune activation? Have we even talked about immune activation? You could use GFAP, which is a marker for astrocytes, but is that a true immune cell? What is the immune cell we talked about today? Because sometimes that's how I mix up the questions. So microglial cells, so you might think of what protein do I find in microglial cells? Do you know any proteins that you find in microglial cells? Sorry? Receptors, sure, or ABCA1 might be one of those proteins. So think a little bit about that in advance. I'd say this is a good discussion question for all of you to have. And I want some other things just as a heads up for you. I want you to be brief in your short answers if at all you can. Yes, it's an evil laugh, but we're not marking you for how well you write grammatically. I had some great poetry. I think I had a haiku in the last term test. I appreciate it, but you don't really need to write those. Keep it brief. You can use point form, so please do so. I want you to give yourself enough time to go back and check. Remember, there are also bonus questions that are at the very end of the exam. Those are somewhat challenging, so if you find yourself kind of bored and nothing else to do and you have like an hour left on the final exam, why not spend an hour on the 2 bonus questions? Not that you'll need to a whole hour, but why not spend some time there? So it's tradition for me to actually give each of you a gift before the final exam. So I'm going to give you a gift of a couple of things to think about. So shall we continue with that tradition? Yeah. Sure. So do you have your cell phones ready? I'm assuming your cell phones are kind of ready. So So this is one of the multiple choice test questions. You're gonna have to think about it. Some of the answer choices might be more challenging, but here's one that you can think a little bit about. So this goes back to thrombotic lesions. We're looking at potassium release. Which of the following might be an experimental animal model? Very similar to the types of questions that you've already had on your quizzes, which is why I really suggest you go back and examine those questions. So slightly different variation, but very similar to the type that you should expect on your final exam. Do you want another one? Yes, because who argues with DJ Khaled, right? So if you're looking at receptor trafficking in hippocampal neurons, maybe think a little bit in advance of the final exam about what the importance of negative spike timing. So you need to know what negative spike timing actually is. I explained it very briefly to you right now, but you should understand like what that's actually asking. If you really understand what that's asking, am I inducing LTP or LTD? How many of you say LTP? How many of you say LTD? Ignore those people in the front. How many of you aren't sure? Okay. So that's, again, something for you to go back. Think about spike timing dependency. Am I strengthening or weakening a synapse? Am I looking at the same mechanisms as LTD or same mechanisms as LTP? What else should we be looking for? So again, I want you to think a little bit about these questions as they're coming up, and I also want you to think a little bit about, for example, the things that we talked about today in class. So I mentioned to you about ApoE, and I want you to think a little bit about ApoE within the brain, and I want you to think back to those questions that I asked you about therapeutics and what sorts of things might we want to enhance when we're looking at things like ApoE. And again, it's one of those things that when you're leaving this course, I hope that if you hear about ApoE2 or ApoE3 or ApoE4, that you'll have a better understanding of how that works, the cells that are involved, and why we really worry about all those different things. Do you have any questions on any of this before we go on? Any questions? No. No questions. Okay. So again, just a quick reminder that tomorrow I do have some office hours around, so you can email me. There's still some times that haven't been booked up. If you come with a group, that would actually help me an awful lot because I don't have to keep explaining myself all the time, and it'd be really useful. I want to thank you for being an amazing group of students and being in this class. I'm really proud of all the work that you've done. There's one other tradition that we normally have in our course, which is to take a quick picture outside. I won't keep you very long, but if you follow Dana, we'll arrange ourselves right outside of Sid Smith. She's standing there at the back. I promise we'll be over and done with quickly. Thank you so much. I didn't ask if there were any questions online. Okay, so one other thing. Can you send me a quick reminder to post all of this online for you, minus pictures of my grandfather for personal reasons? Okay, please go out. Make sure that you get in there for a picture.
Are there any times that you're available to